So good afternoon, and I'm very pleased to welcome you to the Annual Review Prize Lecture of the Physiological Society. The Annual Review Prize Lecture is the highest award that our society bestows on an outstanding physiologist who has made enormous contributions to discovery within our discipline. And this year's recipient is Professor Gero Miesenboeck, the Waynflete Professor of Physiology at the University of Oxford, Fellow of Magdalen College, and he has been awarded the lecture for discovery of optogenetics, which has revolutionized our understanding of biological function. Gero Miesenboeck was the first scientist to use light to control the electrical activity of genetically targeted neurons in both cell culture, in an outstanding paper in Neuron in 2002, and the behaving animal, another paper in Cell in 2005. These were seminal discoveries that created the field we now know as optogenetics, the method of choice to understand how cells communicate with one another, not just in the brain, but in many other biological systems, including the immune system, the cardiovascular system, and cancer. Pre-Miesenboeck, neuroscientists used electrodes to stimulate often ill-defined anatomical regions within the brain. These interventions activated multiple populations of cells whose roles could not be distinguished further. Although informative for delineating the large-scale organization of neural pathways, electrical stimulation really lacked the precision to link activity in specific types of neurons causally to behavior. Gero Miesenboeck is a native Austrian. He completed his medical degree at the University of Innsbruck, spending a little bit of time in Sweden as well. He was just mentioning to me before, doing some medical training there. When he graduated with the top degree from his medical school, he went to the United States to work with the Nobel laureate, James Rothman, in 1993 to take postdoctoral training at Memorial Sloan-Kettering Cancer Center in New York City. From there, he evolved into a faculty position before moving to Yale University as an associate professor on faculty. And we were very fortunate in Oxford to recruit him to the Waynflete Chair in 2011, and he was the inaugural founding director for the Center for Neural Circuits and Behavior. He's a member of EMBO, he's a fellow of the Academy of Medical Sciences, fellow of the Royal Society, member of the Austrian Academy of Sciences, the German Academy of Sciences, and Academia Europea. He has won numerous prizes, so I won't embarrass him by reading all these out, but in particular, I would like to highlight the Brain Prize in 2013, the Massry Prize in 2016, the Rumfeld Prize in 2019, the Warren Albert Foundation Prize in 2019, the Shaw Prize in 2020, and more recently, 2 weeks ago, the Horowitz Prize with Karl Deseroff and Peter Hagelman. What I would say about this last prize is that this is a very powerful prognostic indicator for the Nobel Prize, with over 50% of its recipients receiving that particular honor. It is often argued that the best way to gauge someone's contribution is to ask, where would the field be if that individual was not present? Gero Miesenboeck has single-handedly ushered in a new era of neuroscience. So please join with me in welcoming the 2022 Annual Review Prize Lecturer, Professor Gero Miesenboeck.
Thank you very much, David, for this kind introduction and invitation to give this prestigious lecture. It's a true honor and a privilege to be here. In fact, the privilege is so large that I decided to share it with my doppelganger. Dr. Gero and I have more in common than just the first name. He's also a scientist, a mad one in this Japanese comic called Dragon Balls. He strives for world domination, just like I do. And if you look carefully, you can see that his skull has been replaced with a transparent plexiglass dome so that genetically targeted brain circuits can be controlled with light, using opsins or light-gated ion channels as the crucial interface that transduces the electrical command impulses into electrical activity in the brain. Now, what motivated the invention of optogenetics more than 2 decades ago was the idea that a technology like this would open 3 doors for experimental neuroscience that had previously been locked. The first, as David alluded to in his introduction, was the ability to pinpoint the neuronal causes of cognition and behavior with a precision that was previously unattainable. During my postdoctoral years with Jim Rothman, the mantra that got drilled into us on a daily basis was purify and reconstitute. So disassemble and put things back together. This is the way a biochemist proves that a certain molecule is crucial in a biochemical pathway. So when I started my own lab, I began to think what would be a neurobiologist's equivalent of the biochemist's purification and reconstitution approach. And of course, the equivalent is to metaphorically purify the electrical activity patterns that underpin our mental lives and play them back into a nervous system, and in this way reconstitute perception, action, thought, memory, emotion. The second locked door that I thought optogenetic could open was to facilitate the search for neuronal connections between molecularly defined elements. Imagine that instead of a tedious pairwise search with electrodes, you could simply record from one genetically identified postsynaptic target and raster all presynaptic partners of one particular kind. And the third locked door was the understanding of neuronal mechanisms. As we all know, if one has a hypothesis and wants to find out whether that idea is right or wrong, the only way to do so is through targeted intervention. So for much of the rest of this lecture today, I will focus not on the technology, but on a biological problem in which optogenetics has indeed opened all 3 of these doors for us. And that problem is the neuronal control and biological function of sleep. Sleep is one of the great biological mysteries. I think it's fair to say it's the only ubiquitous behavior whose purpose we don't understand. Sleep carries significant risks and huge opportunity costs that must be offset by some kind of a benefit, but we really don't know what that benefit is. As somebody has put it very provocatively, if sleep didn't fulfill an absolutely essential purpose, it would be the biggest mistake evolution ever made. Now we are trying to get at an answer to this question by studying the neural systems in the brain that regulate sleep and waking. And it's widely thought that there are 2 of these controllers which are symbolized on this diagram by 2 different forms of oscillation. The sine wave symbolizes the circadian clock, which oscillates in synchrony with predictable recurring changes in the external world. As such, it's a purely adaptive mechanism that makes sure that animals do their sleeping at the time that suits them best. But understanding the clock, I think, cannot speak to the deeper question of why we need to sleep in the first place. The answer to that question will come from understanding the second control system, the sleep homeostat, which is symbolized here by the sawtooth oscillation that's superimposed on the sine wave. Now we know a lot about the function of the circadian clock, and this is really the Rosetta Stone that broke the problem open. The discovery more than 50 years ago by Seymour Benzer and his graduate student Ron Konopka of fruit flies whose circadian clocks ran abnormally fast or slow. And from this discovery then flowed over many years and through the work of many different laboratories, a pretty complete molecular, cellular, and systems understanding of circadian timekeeping. This slide, in contrast, and it's an overstatement, but perhaps not too crass an overstatement, summarizes much of what we currently know about the function of the sleep homeostat. And my goal for the next half hour or so will be to try and draw at least a few outlines on this blank canvas. Conceptually, it's clear what the homeostat is. It is what an engineer would call a relaxation oscillator. It's a system that exists in two modes, a fill and a discharge mode, that correspond to the states of waking and sleeping. The homeostat fills with something while we are awake, and we don't know the physical nature of that something. And when that something has risen to a certain level, the system tips into discharge mode. The sleep pressure that has built up is physically causing the system to switch. And during discharge mode, during sleep, that built-up sleep pressure is relieved, and the cycle begins anew when we wake up in the morning. Now, my story also begins in fruit flies a little more than 10 years ago with the discovery by a former postdoc in the lab, Jeff Donnelly, when he was actually a graduate student with Paul Shaw at Washington University in St. Louis, of neurons in the brains of fruit flies whose activity powerfully induces sleep. You see these neurons here labeled with GFP. There's about 24 of them, 12 in each hemisphere. So it's a relatively small minority of all neurons in the brain. They are marked here by a promoter enhancer element called R23E10. So whenever you see that combination of letters and numbers, you know that a genetic manipulation is targeted to these neurons. Now, optogenetics has allowed us to demonstrate the sleep-inducing activity of these neurons, and together, Jeff and I were then able to demonstrate that they have nothing to do with the output of the circadian clock, but instead they convey sleep homeostatic signals. And this is how we do these experiments. We head fix a fly to a custom-built mount, and we let it run on a treadmill that allows us a simple behavioral readout of wakefulness. When the ball is spinning, the fly must be awake since there's no known cases of somnambulance in flies. What you can see is that we've programmed the population of sleep control neurons with a light-gated ion channel in this animal, and we have inserted a patch pipette into one of these 24 neurons to record its activity. And here you can see what happens during an experiment lasting half an hour. The sleep control neuron that we are recording from begins in a state of low activity, and the fly is moving along fine. At about 3.5 minutes, we switch on the lights. The neuron starts to spike vigorously, and all movement quickly ceases. Shortly before 20 minutes, we switch the lights off. The activity of the neuron once again drops and movement resumes. And a few minutes later, we switch the lights back on, and we toggle the animal back into sleep. So it seems that we have isolated a switch in the brain that allows us to force the fruit fly into and out of sleep. Now, the existence of these sleep-inducing cells in the brains of flies has a remarkable parallel that actually provides a historical reminder of the last great pandemic that swept the globe more than 100 years ago. The influenza pandemic at the end and in the aftermath of World War I. And the young neurologist in Vienna, Konstantin von Economo, described a new disease during that period, which he called encephalitis lethargica. These were patients who actually survived a severe bout of influenza and afterwards were either somnolent or incurable insomniacs. And as these patients began to die off in the coming years, von Economo teamed up with neuropathologists and discovered that in the midbrain there is two regions where lesions in these patients clustered. And depending on whether they were hypersomniacs or insomniacs, either an area that's now known as the ventrolateral preoptic nucleus of the hypothalamus, which is shown here in magenta, and is the sleep-inducing area, were affected, or an adjacent area consisting of the tuberomammillary nucleus and the VTA. Now, von Economos' discovery showed that sleep is not simply the brain falling silent, but it's an active process that is actively induced and actively maintained. And it also showed that there is a small dedicated circuitry in the brain that's responsible for the induction and maintenance of sleep. And we believe that the neurons that we study in the fly are the equivalent of the ones that von Economy first identified. Now, our recordings from these sleep-inducing neurons in the fruit fly revealed that their biophysical properties can vary dramatically. Some neurons, like the one shown here on the left, spike vigorously if you inject depolarizing current, whereas others, like the cell shown on the right, don't emit any action potentials at all. It's not just the ability to produce spikes, though, that seems to vary in neurons recorded, in this case, in different flies. It's also their passive membrane properties. If you look at the voltage responses to hyperpolarizing current steps, you can see that on the left, a 5 picoamp-sized hyperpolarizing current step causes a voltage deflection of almost 20 millivolts, whereas on the right, it's less than 5 millivolts. So by Ohm's law, the resistance with which the neuron on the left opposes the injected current is large, whereas the one on the right opposes it with a much lower resistance. The neuron on the left also takes a long time to find its new membrane potential equilibrium after a current step, whereas the neuron on the right settles into the new equilibrium very quickly. Now, this combination of low input resistance and short membrane time constant that characterizes the cell on the right is virtually diagnostic of the opening of a current shunt. And I'll show you in a moment what the molecular carrier of that current shunt is. Now, when we saw this, we thought, well, maybe this is the biophysical mechanism at the core of sleep homeostasis. Maybe these sleep-inducing neurons switch between a state on the left when sleep pressure is high and a state on the right when sleep pressure is low, when you are well rested. And anecdotally, when we manipulated the sleep history of flies by sleep depriving them or giving them a night of undisturbed rest, and then measuring the activity of these neurons, we indeed found that this idea was borne out. But of course, these are just anecdotal observations. They are just sampling from a population of flies and from a population of neurons. What one would really want to do is actually to take charge of the switch and to show that one and the same sleep-inducing neurons can indeed make these transitions that I have just described. Now, in order to do that, one would, of course, need to know a signal that naturally operates on the switch. Now, what might such a signal be? In the first experiments that actually demonstrated optogenetic control of animal behavior, my then graduate student Susanna Lima at Yale focused on several types of neuron in the fly's brain, including dopaminergic neurons. And she recorded the movement trajectories of flies in circular arenas before and after switching on the dopaminergic system for 2 minutes each. And you can see here the strongly arousal-promoting effect of dopamine. These are the same flies before and after switching on the dopaminergic neurons. Now, that dopamine would be an arousing signal is not surprising. It's true also for us. All psychostimulants, cocaine, amphetamine, of course, as you know, work by blocking dopamine reuptake at the synapse. And there indeed seems to be a dopamine delivery system in the fly's brain that's custom tailored to provide that arousing dopamine to the sleep control cells, because the two cell types shadow each other incredibly tightly. So of course the question arises, are they connected? And optogenetics is just the tool to allow us to answer this question. We can record from a sleep-inducing neuron while photostimulating dopamine release from the population of dopaminergic neurons. And what we saw during these experiments was that we could actually drive the switch that I showed to you before just by applying dopamine. So the very same neuron would switch from the electrically active on state to the electrically silent off state. And if we held the recording long enough, would spontaneously return to the electrically active on state. It's not just the ability to generate spikes, though, that varies. You can see we've also modulated the passive membrane properties, the input resistance, and the membrane time constant. These changes occur on time scales of a few minutes and are therefore way too fast to be accounted for by transcriptional changes. Instead, they must involve modifications of the existing channel repertoire in these neurons. So what are the channels that are affected? Knowing the ability, or having the ability to control the switch through dopamine allowed us to record currents while we performed the same manipulations and documenting changes that were elicited by dopamine. And these recordings quickly revealed that potassium currents were at the heart of this change in excitability, and specifically two types of potassium currents. A voltage-gated potassium current, which was high in the electrically active on state and became down-regulated by dopamine. So as the neuron reduced its activity, this voltage-gated A-type current dropped and then rose again when the neuron spontaneously reverted to the electrically active state. And these changes were mirrored by opposite changes in a voltage-independent non-A-type current, a leak current, that was low in the electrically active state, became dramatically upregulated in the off state, and then again reverted to its basal level in the electrically silent state. Now, the molecular carriers of these two currents are two potassium channels that we identified through targeted RNAi-mediated interference screen. The A-type current is carried by the prototypical voltage-gated potassium channel, the Kv1 channel shaker, whereas the leak current is carried by a 2-pore domain potassium channel that we dubbed Sandman, following the tradition of the Drosophila field to name genes and their products after the mutant phenotype. Flies that are mutant for Sandman cannot switch their sleep-inducing neurons off. And so they literally doze away their existences. They sleep 23 and a half hours a day. And it's the absence of the increase in Sandman when the arousing dopamine arrives that, of course, accounts for the phenotype. And the 2-pore domain potassium channel Sandman is, of course, the molecular carrier of the current leak that I've shown to you before. So we've reached a stage where we can take a rather vague and diffuse question that we started out with, such as what is the function of sleep, and give it a tractable, biophysically precise form, namely what regulates the function of these two ion channels, shaker and Sandman, in these neurons. Any sleep-inducing signal that, by definition, alters the electrical activity of the sleep-promoting cells must signal through one or the other or both of these ion channels. So we might be able to find a molecular signature of the biological process that these sleep-inducing neurons monitor by answering this question. I will focus exclusively on our work on shaker. There has also been significant progress in understanding the regulation of Sandman, but in the interest of time, I just focus on shaker. Now, shaker is actually only one subunit of a 2-subunit ion channel complex, shown here in Rod McKinnon's beautiful crystal structure. Shaker is the gray portion, the ion-conducting, membrane-spanning portion of the channel. And appended to that, like the gondola under a hot air balloon, is a beta subunit shown here in blue. Now, these beta subunits have been mysterious since the first sequences were cloned some 3 decades ago, because the sequences revealed a surprising homology to enzymes, specifically to aldo-keto reductases. And the crystal structure bore out that sequence homology because McKinnon clearly saw an active site, and sticking into the active site is the redox end of a cofactor shown here in red, which is NADPH or NADP+. But what makes this association between a voltage-gated ion channel and an enzymatic subunit so particularly mysterious and baroque is the fact that the enzyme actually looks like a really subpar catalyst. If you look carefully on top of the red NADPH that's bound deep in a cleft, there's a tryptophan residue that sits on top of that cleft like a latch and locks the cofactor firmly in place. Now, if catalysis is your goal and you want to turn over lots of substrate in a short period of time, you would not want to lock one of your reactants in place. That would choke catalysis. So what is this whole assembly? Is it a voltage-controlled oxidoreductase? Is this the reason why the beta subunit is associated with the ion channel? Or is communication in the other direction? Does the beta subunit somehow control the function of the channel? We believe that it's both, and that it's actually the dialogue between the gate that regulates potassium flux through the channel and the active site of the beta subunit that's the fundamental accounting principle at the heart of sleep homeostasis. Specifically, we've done many experiments that suggest that the potassium channel beta subunit is a sensor of how efficiently the mitochondrial electron transport chain uses the electrons that are fed into it as fuel. This animation shows a very simple version of the mitochondrial electron transport chain in the inner membrane. You see the three main complexes, 1, 3, and 4. Complex 1 receives electrons from NADH, which in turn derives them from the Krebs cycle mostly. And they then get passed down to complexes 3 and 4. Complex 4 is the so-called terminal oxidase, takes 4 electrons and transfers them to molecular oxygen and thereby reducing it to water. Now, all 3 of these complexes are also proton pumps. They pump protons from the matrix into the intermembrane space, and these protons then flow down the electrochemical gradient and power the turbine that you see spinning on the right. This is the rotary ATP synthase. What you see here is a mitochondrial electron transport chain that's in perfect equilibrium, where every electron that gets fed into at complex 1 gets actually used ultimately to power ATP synthesis. And in these conditions, we think, and we have shown, the beta subunit is mostly in the NADPH bound state, so the cofactor is reduced. But there are several conditions that actually lead to an imbalance in mitochondrial electron transport. And this is probably the most common. If the supply of electrons exceeds the demands of ATP synthesis, the extra electrons that get stuffed into the transport chain at complex 1 have nowhere to go, they stall, and they start to spill from the rims of the ubiquinone pool, complexes 1 and 3, reacting directly with molecular oxygen, producing oxygen-free radicals, and that then, we think, leads to the oxidation of the bound cofactor, so NADPH gets oxidized to NADP+. So in this way, the potassium channel knows how efficiently the mitochondrial electron transport chain is operating. Now I'll show you, just since this work is published, 2 crucial pieces of evidence that implicate the mitochondrial electron transport chain and the potassium channel beta subunit as the crucial elements, the sender and the receiver of this metabolic message. We've done many different experiments and manipulations that implicate mitochondria, but probably the most powerful is the expression of an enzyme called alternative oxidase, or AOX, which is present in many plants and some microorganisms. And these organisms have bifurcated electron transport chains. in contrast to our strictly linear transport chains where complex 4 is the only terminal oxidase. So they have a second terminal oxidase, the alternative one, that actually taps into the ubiquinone pool and siphons off electrons that would otherwise be in surplus and transfers them directly to molecular oxygen, produces water, and therefore prevents the production of reactive oxygen species. It's, if you wish, a pressure relief valve that releases the extra electrons if the transport chain overheats. Now, if we introduce this overflow valve into the transport chain of just these sleep control neurons, you can see that we cut the need to sleep dramatically by 7 to 8 to sometimes 9 hours a day. Now, this signal, we think, that's generated in the electron transport chain is actually sensed by the potassium channel at the plasma membrane. And of course, either mutating shaker or the beta subunit creates insomniac flies, but probably the strongest experiments that we did to actually show the crucial involvement of the oxidation state of the beta subunit was once again an optogenetic strategy, but this is now an optogenetic tool that does not control membrane voltage, but it controls redox chemistry. It was a tool that was developed by the late Roger Chen as a genetically encoded contrast agent for electron microscopy. It's called mini superoxide generator, or MiniSOG. And we anchored this protein at the inner leaflet of the plasma membrane through a post-translational modification, and then tested whether illuminating it in the vicinity of the potassium channel beta subunit, which in flies is called hyperkinetic, or HK, could actually acutely induce sleep. And the answer is yes. So you see in these actograms the movement trajectories of populations of flies during 9 minutes of illumination. And if you look at the third graph from the left, these are the experimental flies. You can see that they spend more time asleep, they enter longer and more frequent sleep episodes. But this transduction of oxidative stress into sleep can be blocked completely if we simultaneously remove through RNAi the hyperkinetic protein, the potassium channel beta subunit. That's the diagram all the way on the right. Now, having this MiniSOG trick to manipulate the redox state of the cofactor also allows us to do some within-cell physiology and compare the biophysical properties of these sleep-inducing neurons before and after the induction of oxidative stress. And we see that activating MiniSOG through light increases the action potential frequency. It steepens the input-output relationship of these neurons. And what we think the underlying mechanism is a slowing of the potassium channel, the A-type current inactivation time constant, which you see on the right. Now, how would potassium channel inactivation be linked to spike rate? The intuition and some modeling suggests that if you have a slowly inactivating potassium channel in a tonically or persistently active neuron, then each action potential will push a smaller fraction of the channel population into the inactivated state. So you always have a powerful repolarizing force that allows you to return the membrane potential to its resting level after each spike. And in this way, you can fire at higher rates, just as we observe. But there was one issue that always bothered me a little bit about this model of communication through redox chemistry between the inner mitochondrial membrane and a potassium channel at the plasma membrane. It's an awfully long distance to bridge for a highly reactive, immediate, very, very short-lived molecule. So could there be a potential intermediate that actually is generated and that fulfills, if you wish, the role of second messenger? And we think there is, and the intermediate are some of the most abundant and vulnerable targets of reactive oxygen species in cells, and these are the polyunsaturated fatty acids of membrane lipids. We believe that the oxygen-free radicals that are generated locally attack these membrane lipids, and it's well known that an oxidative attack on the double bonds of these polyunsaturated membrane lipids leads to a series of rearrangements and scissions that produces fragments of the polyunsaturated fatty acids, such as the aldehyde 4-oxo-2 nonenal. So you may remember that the potassium channel beta subunit is an aldo-keto-reductase. So its native substrates, although they have not been conclusively identified, must be carbonyls, and this would be one candidate, a molecule with the right chemical functionality to fit into the active site of the beta subunit, and then be reduced to the alcohol and being one part of the redox couple that drives simultaneously the oxidation of NADPH to NADP+. So we tested some aspects of this idea, relying on voltage clamp measurements and using the inactivation time constant of the A-type current as our index of whether the potassium channel population in this in the sleep control neuron that we are recording from is in the oxidized or in the reduced state, because we know that if NADPH is bound, the current is fast inactivating. If NADP+ is bound, the current is slowly inactivating. So we included in the patch pipette our candidate molecule for oxo-2 nonenal and waited 10 minutes, and you can see that indeed we see a conversion of the channel population to the slowly inactivating form, because you see that the fast inactivation time constant is rising. Now, this conversion happens only if the neuron is equipped with a catalytically competent beta subunit, with a catalytically competent hyperkinetic. If we infiltrate the channel with a catalytically defective protein that has a single point mutation that destroys its catalytic activity, the rise in the inactivation time constant does not occur. The rise in the inactivation time constant is also stable for a very, very long time, for essentially as long as we can hold these difficult voltage clamp recordings. So this would almost suggest that this is sort of a biochemical memory cell, that each of these beta subunits, because it's catalytically so inefficient, holds on to the oxidized cofactor and sort of forms a stable memory of the encounter with an oxidizing substrate, such as 4-oxo-2 nonenal. But of course, in this case, our patch pipette is full of potential substrate, so what we could be observing in this condition is, in fact, not a stable biochemical memory, but continuous turnover of substrate. But our optogenetic minisoc strategy gives us a way to actually initiate and terminate the production of the putative substrates, because we can obviously switch the lights off, but we can't remove the 4-oxo-2 nonenal. So repeating the same experiment, you can see that the minisoc also elevates the inactivation time constant to the same extent as the provision of the synthetic molecule. And if we then switch off the lights and again wait for 20 further minutes at resting membrane potential, clamping the neuron at -60 millivolt, you see that this elevation is stable. So indeed, the beta subunit is sort of a lasting record of lipid peroxidation. Now, if you think back to the normal physiology of these sleep-inducing neurons, they alternate, depending on sleep pressure, between high and low electrical activity. So this would conform to a sleep control neuron that's in a state of low activity and an animal that's awake, where lipid peroxidation happens and is registered in the form of this biochemical memory. But once sleep is induced and electrical activity resumes, you would expect that that sleep pressure somehow needs to be dissipated, and that therefore perhaps it's the electrical activity that in turn is controlled by the oxidation state of the beta subunit that drives cofactor exchange. So we tested that for both the chemical manipulation, the inclusion of the 4-oxo-2 nonenal in the patch pipette, and the optogenetic manipulation, and you can see that if we simulate spiking by depolarizing the neuron at 10 Hertz for 20 minutes, we can drive the inactivation time constants back down to baseline. So what we then think this potassium channel assembly is, is a biochemical memory cell consisting of a membrane-spanning, voltage-sensitive and ion-conducting alpha subunit and a redox-sensing and catalytically poorly active beta subunit. But these two molecules are in dialogue and together then form this biochemical memory cell that actually resembles a semiconductor memory cell in dynamic RAM, like in our computers or cell phones, which also have two components. These dynamic random access memories have a storage capacitor that holds 1 bit of information and an access transistor that regulates whether the storage capacitor can be updated, whether the information that's stored there can be written or read. I've drawn this superimposed on the potassium channel because we think that the beta subunit, in fact, corresponds to the storage capacitor, except that it doesn't hold the 1-bit information in the form of an electrical charge. It holds 1 bit of information in the redox potential of the cofactor NADPH or NADP+. That information sits there for a long time, although it needs to be periodically refreshed because there must be some kind of spontaneous turnover, just like the information held on the storage capacitor must be periodically refreshed. But the main device that generates access to the storage capacitor is the voltage across the access transistor. In the semiconductor and in the potassium channel, it's of course the voltage across the voltage sensors of the alpha subunit. So the two pieces of information that this device integrates in electronics called the word line and the bit line correspond in our system to the electrical activity and to mitochondrial metabolism. So the information that's being written and read is the redox signal that comes from the mitochondria, and whether the information is stored depends on the electrical activity of the neuron. The cell is in write mode if the activity is low, and the information is destructively read. That means used and erased when the electrical activity is high. So we've reached a stage then where we can replace this conceptual animation that I began with a mechanistic picture. We have a neuron that can transition between two extremes of states, an off state that you see here where it's electrically silent, and where the Sandman channel, which is the channel shown here in yellow, is present in the plasma membrane. We know, but I haven't shown you the evidence, that these Sandman channels actually are regulated by membrane trafficking. So they are endocytosed and reinserted into the plasma membrane, and we believe that it's the position of Sandman within the cell that determines whether the container in our conceptual animation is in fill or discharge mode, whether it's upright or tilted. When Sandman is endocytosed, or if Sandman is present in the plasma membrane, as you see now, we would be in fill mode, and the potassium channel beta subunit gradually fills with NADP+ as the neuron undergoes oxidative stress. Once the system tips, the voltage-gated rearrangements of the channel allow us to exchange the cofactor and discharge the accumulated sleep pressure. Now, I began with the premise that understanding the signals that regulate these two ion channels in these neurons could offer us a potential clue to sleep's enigmatic purpose. So is there indeed a causal connection between mitochondrial energy metabolism and what sleep is good for? Or are these sleep-inducing neurons simply using this mechanism that I've described as an hourglass, a mechanism to measure time on timescales that are much longer than an individual action potential? Now, how could we possibly answer this question? We've made one attempt, and that relies on two genetic manipulations that are targeted to the brain. The first one is our alternative oxidase, which you are familiar with, which acts as an overflow valve in the electron transport chain and causes a significant sleep deficit. And the other one is the removal of the potassium channel beta subunit, which also causes a sleep deficit. And here you can see that the sleep deficits in both cases are substantial, but AOX causes an even more severe insomnia than the mutation of the potassium channel beta subunit. So we have severely insomniac flies, but the root cause of insomnia is quite different in the two conditions. If our hypothesis that sleep is somehow necessary to offset lipid peroxidation or oxidative stress, then the introduction of AOX should cure that problem at the source. Whereas the removal of the sensor of the Kv beta subunit should in fact only make the system unaware of the problem and therefore make the situation worse. So we decided to take our 2 or 3 groups of insomniac flies and look for some of the classical adverse consequences of chronic insomnia. And there are 2. The first is cognitive impairment. So we tried to teach these flies a simple lesson, Pavlovian conditioning of an odor with an aversive electric shock. And you can see that the insomniacs that lack hyperkinetic, shown here in red, indeed have a memory deficit. The other classical adverse consequence of chronic sleep loss is a shortened lifespan, which hyperkinetic mutants also show, premature aging. But our AOX overexpressors do not have these adverse consequences, despite their sleep deficits being more profound than those of the others. You can see they learn just fine, and if anything, they lead a slightly longer life than their wild-type controls. Now, with that, I've almost reached the end of my talk, but I would like to bookend the lecture. I've begun with optogenetics, and I would like to return for just 1 minute to the Stone Age of optogenetics, to the days when we first began to think about the potential uses and the implication of a method like this. And during these days, I became aware through a citation alert to another of my publications that I wasn't the only scientist who had seen the need for a technology like this. In a paper entitled The Impact of Molecular Biology on Neuroscience that was published in the millennial issue of the Philosophical Transactions of the Royal Society, Francis Crick stated that the next requirement for understanding the brain is to be able to turn the firing of one or more types of neuron on or off in a rapid manner in the behaving animal. The ideal signal would be light. This seems rather far-fetched, but it is conceivable that molecular biologists could engineer a particular cell type to be sensitive to light in this way. So Crick clearly saw the need, but he didn't spell out the way. He didn't give any instructions of how to do it. So when we had the first experiments that turned this far-fetched possibility into a reality, and these are these experiments, these are hippocampal neurons grown in culture and modified to express an opsin and several auxiliary genes, you can see that as we record the membrane potential of these neurons in the dark, it hovers around resting value, but as soon as we switch on the lights, the neuron responds with a volley of action potentials. So when we had these experiments, I decided to send Crick a preprint of the paper. And if you've read this wonderful book called The Eighth Day of Creation by Horace Judson Freeland, which recounts the early days of molecular biology, you know that Crick was an enthusiastic letter writer who carried out a vast correspondence, taking intense interest in other people's work. And his hallmarks as a correspondent were twofold. He always mixed encouragement and constructive criticism, and that's exactly what I got. So Crick wrote back and said that he had read the paper I'd sent him with great interest and was excited to see that the system already worked, at least to some extent. However, he realized, as I did, that it still needed improvement and that this will take further work. Unfortunately, Crick didn't live to hear how not just our experiments progressed, but also those of others who later in increasing droves joined the field. But the improvements have come, most importantly due to the molecular identification of channel rhodopsin by Peter Hegemann, which happened after these early studies. But I think as a consequence of these combined efforts, it's fair to say that the way we neuroscientists approach our problems has changed. With that, I am at the end of my lecture, but I would like to briefly acknowledge the people responsible for the work. Boris Semmelmann was the postdoc who built the first optogenetic actuators. Susanna Lima was the graduate student who used them in an intact animal. Jeff Donnelly and Diogo Pimentel were two postdocs who started our work on sleep. Anissa Kempf, another postdoc, and Michael Song, a graduate student, worked out the connection between mitochondrial energy metabolism and the potassium channel beta subunit. Olaf Rossmann did the studies on the biochemical memory of the beta subunit, and Alina Krebers the studies of sleep, longevity, and memory. I should say that the current crew on this slide is lined up to the left, and the alumni are displaced by one tab stop to the right. Now that's really it. Thank you. Thanks.
Gero, thank you for an outstanding lecture, thought-provoking, seeing originality of discovery, which is what our discipline is all about as a discovery science, and I think you've demonstrated that in spades today. So I would just like to present you with two rather humble awards here. The first one is from the Physiological Society, which is a small memento for the prize lecturer, and the second is from our friends in Denmark, which I've been told is not a duck but a bird. And this also is a great acknowledgement to your terrific lecture this afternoon. So thank you very much. Thank you. I think we've got to, you hold that, and we've got to get some pictures.